Okay, so I think we can continue now with, so this time it will be Clarisse. So for this second part about lab work, DNA extraction, PCR, bioinformatic analysis. And so, yes, during the talk, I think do not hesitate. If you really miss things, raise your hand, and then we can you can stop. Is it okay like that, Clarisse? Yeah.
Yes.
Yeah. Did you take a coffee?
It will be after.
There was coffee? No coffee, my dear. Clarisse, be strong.
Should be.
Okay, so thank you very much.
Thank you. So here we are after lunch for the second part of the Metabarcoding workflow. Well, we will go through all of it and see particularly the lab work and the sequencing technology into details and the bioinformatic pipeline. So indeed, I will go through all these steps. Don't hesitate if you have any questions. I think I will take a moment at the end of each steps for before going to the other ones, as it will change a lot. So the first thing I want to mention is that in a metabarcoding workflow, you are using DNA, and DNA is invisible. And at one time, you amplify this DNA, so it can be quite sensible, and you have to be aware of putative contamination. And you need to work in clean conditions and have in mind that you don't want DNA from one sample to be mixed with DNA from another sample because you won't have good results at the end. So, of course, you will be more aware of that if you are studying ancient DNA, where you have a very few of this DNA, than studying a diatom biofilm, for instance, where you will have thousands of cells in a very small area. So by clean conditions, I mean use clean material for the sampling, and also the DNA extraction and PCR steps. It's better if they are done under sterile environments, and you will add also negative control at each steps to see, okay, do I have clean regions that works with my workflow? So for the sampling, you can use either sterile devices, for instance, for phytoplankton, but I will describe it a little bit later. We use Sterivex filters that are provided already without DNA. And we use also syringe that are packed, unit packed and without DNA. But you can also, for the bucket used to sample water, you can just remove DNA using various solutions like hydrogen peroxides, hydrochloric acids, bleach, and so on. And another thing that is important is that DNA is a molecule and can degrade. We were speaking about that for the conservation of ancient DNA. So if you want to preserve your DNA from your sample, it's ideal to put it in a low temperature where the DNA won't degrade that much. But there are also other solutions, because you won't have a freezer near you during the sampling at any time. So you can add other solutions that will stabilize the DNA, like buffers, or use ethanol, for instance, that can also preserve DNA. So now I'm gonna, maybe if you had some questions on this part, I don't know. Yes?
Can you explain a bit this negative control?
Yeah. So for instance, so they will be present in, they can be present in different steps of the metabarcoding. For instance, in the sampling, it can be just to take a device that you use for the sampling, for instance, a bucket or a filter, and then you can try to extract DNA from that filter that wasn't used in the field, and you can see if you already had DNA at the beginning from this filter. It can be something like that. Also for DNA extraction, you put a samples that is supposed to doesn't have DNA in it, and you go through the protocols. And at the end, you look if you have DNA. There is a problem because you shouldn't. It's this kind of.
So for the field, for example, znači to take filter and put a buffer and proceed with this. Thank you.
Yes.
So sterile water, for instance.
Or sterile water.
Yeah, you got the filter sterile water and look.
We can include it.
That it's present in every sampling because it can be quite heavy to always extract DNA from negative control, but it's nice to verify sometimes that everything is working well.
But you have to think that the filters are very expensive.
Yeah.
So
Yeah.
one of these Sterex filters is maybe $16. So we have to be aware not to use too many.
Yeah. It can be used if you are regularly sampling. It can be used every 3 months or every year in case to be sure that everything is fine.
Okay, thank you.
So if everything is good, we can go through DNA in good quantity and good quality of the targeted organisms we are interested in. So in your sample, in your soil sample, in your water sample, DNA molecule will be associated with a lot of various molecules from the matrix, if it's soil or if it's a biofilm. It will be also associated within the cells with within the DNA molecules, you have those DNA. So basically, during DNA extraction, you want to get rid of all of this and only keep the DNA molecule. Also, in a given sample, you can have what we call inhibitors that will make the amplification of the barcode that won't make it work. It won't work. It's really something you don't want because it's a mandatory steps for your metabarcoding workflow. So what are the main steps of DNA extraction? The first one, you lysate the cellular membrane in order to release all the cellular content in your sample. The second step is to separate your DNA molecule from all the other things you don't want. And the third one is to elute DNA, that is to concentrate it. In that way, you will have a good quantity of DNA in a small volume of water, for instance. So all these 3 steps need to be adapted to the matrix of your sample. You are not going to extract DNA the same way from a soil sample, from a water sample, or if you are targeting tissue of animals. Also, it needs to be adapted to the organisms you want to target. It won't be the same complexity to lysate a bacterial cell as compared to a diatoms or a copepoda, for instance. And it needs to be adapted to the questions. If you are interested in extracellular DNA or intracellular DNA, etc., it's at these steps that you will manage that. So you have 2 options for DNA extraction. It's either using organic solvents for solution for the extraction, which is called the phenyl chloroform protocol, or you can use commercial kits that they present the advantage that they can be already optimized for different matrices of samples. So the first part, the lysis of the cellular membrane. For that, we have plenty solutions that can be grouped in 3 main categories. The first one are the physical lysis of the cellular membrane, where you can break the cells either with mechanical forces. Okay. Should I? Okay. I'm gonna remove all this. Okay. So for instance, you can put your samples in these tubes you have on the bottom of the slide, bottom left, that present small beads, and then you agitate your sample with those beads using a vortex, for instance, with high speeds, and the movement of the beads will break down the cells. You can also use freeze-thaw cycles or other techniques. Then you have another option is of using chemicals, because your cellular membrane is composed of lipids. If you use detergents, for instance, it will destabilize the lipidic membrane and release the cell contents. And the last way to break the cell membrane is to use enzymes, because microbes and eukaryotes already try to kill each other using these enzymes. So it's also an option. You can combine different type of lysis in order to be to optimize and be sure that you extract DNA. The second part is to separate DNA from all the other things. So for that, we use natural properties of DNA. That is, it is hydrosoluble and it is negatively charged. Agnes showed us the structure of the DNA molecule, and in the phosphate deoxyribose backbone, you have this negatively charged oxygen. So the first way to separate DNA from everything is to use silice columns that are used in kits, for instance, in commercial kits. So under certain conditions, with low pH and high salts, the DNA, these columns will be positively charged and the DNA will bind to it. Then, so it looks like this. You have this tube with a silice membrane here, and then you put your samples with everything, and you do a spin with centrifugation, and all the liquid will go down except DNA because it's bound to the silicate membrane. And then you remove this, and then you can also add washing steps to be sure that you will only have DNA at the end. And when you change the conditions with higher pH and low salts, the silicate membrane becomes negative and the DNA will separate from it. And then you can elute your DNA, putting a small volume of solution. So it is one way that is widely used for separation of DNA from all the other molecules. Another way is to use magnetic beads. It's also present in kits. So magnetic beads are very small iron oxide particles, and they are coated with silica. So also it's the same principle. If you change the conditions, either your DNA will bind and either your DNA will be released. And you can segregate those beads using magnets. And the last way is to use organic solvents, phenylchloroform, where the lipids will dissolve and not the DNA, because the DNA will dissolve in the water. So you have here your lysates with all DNA and proteins, membranes, etc. And then you add this phenylchloroform that will be present in the bottom, and you will find your lipids here. You will find the DNA in the aqueous phase, and in between you will find proteins. And then you just have to take the aqueous phase and put it in another tube. And then you precipitate your DNA with salts and isopropanol, and then it forms a pellet. in the bottom of the tube, and then you can put another solution and have your DNA back. Sometimes, if you're lucky, you can visualize your DNA, but it's when you extract it from very large, like for cultures of cells, you can see it. I never saw it using these techniques. So now you extracted DNA, but basically you have water in the tube and you don't know if you really extracted DNA and if it is of good quality. So for this, we have different techniques to make sure we really extracted DNA. The first one is to use electrophoresis. So DNA is, you put your DNA, your just extracted DNA in the sample wells of agarose gel, and then you apply a current, and the DNA, as it is negatively charged, will migrate to the positive electrodes. And then you can dye this DNA using intercallant dye that fluoresces under UV light. And it looks like this. So here you just have this gel and what you can see after the migration. So you often add a layer with known DNA length that you can estimate what is the length of your DNA. The smaller your DNA, the most far it will migrate during this process. So here in this study, they just put DNA that they just extracted in those wells, and you can have profile like this. So we see that we have a high fragment of DNA on the top, and then you have what is called smear, which is a gradient of different DNA lengths because you have different size of DNA. If you look the first DNA extraction, so those 3 first wells were extracted using the same protocols, we see that we don't have this high length of DNA, but only the smear. And it's because this protocol of DNA extraction might have degraded a lot the DNA during its process. So it's something you can check at this step. Another way to see if your DNA is of good quantity and also good quality is to use the electrophoresis. So it's with this, not the electrophoresis, the spectrophotometer. So with a device that is called nanodrop. And this device works with the absorbance of DNA that is at the wavelength of 260 nanometers. So the higher the peak, the more DNA you will have extracted. And also this device measures the absorbance of other molecules at other wavelengths. For instance, 230 nanometers, you will have proteins that will absorb at this wavelengths. So if you have, and it measures the ratio between the absorbance at the DNA wavelengths and the absorbance at the protein wavelengths. And this ratio is not good. That means that you have a lot of proteins in your DNA extracted. And similarly, this device measures absorbance at 280 nanometers, which is the absorbance for more organic contaminants. So in that way, you can see if you have a good quantity of DNA and a good quality of DNA. Yeah, maybe if you have some questions in this first step, before we go through the PCR.
Was simply to ask you if you had some questions. Oh, sorry.
No. So,
Is it clear? Is it clear for you the gel electrophoresis, for instance, how it's working? Yeah. Okay. So.
Perfect.
So we can continue.
We can continue and go through. So the objectives of these steps is to amplify the barcodes in thousands of identical copies. And it is during these steps that you truly see if you didn't extracted, for instance, inhibitors in your DNA extraction. So we again didn't invent anything. We just used what already exists in nature to amplify DNA. There is a protein called DNA polymerase that its only job is to replicate at the base, really replicates a strand of DNA. And this is used during the cell division, because at the end you want the same identical genomes in one cell and the other cell. So this DNA polymerase replicates your DNA, and that's it. So one thing you have to know is that these proteins cannot synthesize a new DNA strands de novo. It can only add new nucleotides to already existing sequences. It cannot start, it can just go there and then continue to put complementary nucleotides of these DNA strands. So in the lab, what we do is that we artificially separate the two DNA strands, and then we use the primers. Those famous primers I spoke about before lunch, to initiate the replication that DNA polymerase can initiate, replicate the strands. And we use what is called a high fidelity DNA polymerase, that we want it not to make mistakes, because we are interested at the sequence at the end. So we really want a good DNA polymerase. So what are the different steps of polymerase chain reaction? In your in the beginning, you only need your DNA template that you just extracted, then a DNA polymerase, then nucleotides to synthesize a new strand, DNA strands, and then also your primers, forward and reverse. So the first step for the DNA naturation, we do it at high temperature, 95 degrees, and at this temperature, the two strands will separate. Then we put the reactive at a lower temperature that will be optimal for the primers to anneal in those two strands. And then the polymerase can do its job and extend the complementary strands based on the sequence of the first one. And then at the end, you have two new molecules of DNA. And we repeat it a lot of times for 25, 30 cycles. And then it will be exponential. You will increase the number of molecules you got. And those molecules will be at the length delimited by the primers. Something we have to know is that during these steps, we create artificial fragments that do not exist in reality. And this is due because you don't have only one template of DNA from one species, but also because you have multiple species in metabarcoding. And sometimes the polymerase started to synthesize a new strand and stop for some reason. And the problem is that fragment that was newly synthesized with the primers and the little piece of a new fragment can hybrid to a wrong template that was not the same at the beginning. But it can work, and the polymerase continue its job. And then you will have a sequence that is a mix of two true sequence at the beginning. And you will amplify them. They will, the PCR continue, you will amplify them, and you will get them at the end. And then to check if your PCR worked well, you use this electrophoresis I explained you before, and now you expect to have only a signal in a certain length of the amplicon size. So if you have any questions on these steps.
So Julian online asked, does the silicate wall of diatoms is a problem during the DNA extraction step?
It is a very interesting question, and it is, in fact, but maybe, Frederick, you're going to speak a little bit more. But yes, it's a very, very good question. And indeed, I don't know precisely how you get rid of the diatom silicate structure.
Yeah, in fact, we will talk about this in the talk just after you. But yes, we in fact, we tested several kits, several extraction kits and methods, and one was better than the others. And in fact, the cell wall, the silicate cell wall, a big problem when you have lots of fluctuation of diatom, but it's not. I don't think it's a problem in biofilms. Yeah, I don't know if you have other explanation, Agnes. Do you? No. But we are not, for instance, we are not using. I can imagine, for instance, if we would use some diatomite, so this stone made of diatom fructule, which is really like a filter. Yes, here it can be a problem for if you have some DNA lost in this forest of fructules. Yes, here it can be a problem, but not when we are working on biofilms. I don't know if it's
It's the same for clays, for instance, that can also absorb DNA. So you have to add extra steps in your DNA protocols when you work with this kind of samples. But yeah, so the next part is now you have amplified your barcodes, you have those nice bands, and you can sequence those barcodes. So the objective is basically to get the DNA sequence of all your amplified barcodes. And for this, there are different sequencing technologies that exist. So I will just get through this. The first generation was the first one that appeared in the 90s. With a machine called Sanger. With this generation of sequencer, you can have around 100 sequence per run of a size between 400 and 900 bare paste, and with a very small error rates. Then, in the beginning of the 21st century, you have the second generation of sequencers with Roche 454, Illumina, and Ion Torrent. That's really, it's the high throughput sequencing that Agnes was speaking, that we really increased the depth of your sequencing, where you can get millions of sequence in one run, but of a smaller size, and also with low error rates. And the third generation of PacBio Oxford Nanopore, they are very cool because you can have quite a good amount of sequence and with very large fragments of DNA. But the problem is at the beginning, you had very high error rates. Now it's decreasing, but it was not compatible to, when you want to analyze the sequence and see their composition, you cannot get far with such error rates. So that's why the technologies that are currently used for DNA metabarcoding are from the second generation and mostly Illumina technology. So it is this one I will develop more in details. So you can sequence a length of 400 and 500 bare pairs max. And this is because we do paired end sequencing. Why? Because Illumina can sequence small, short sequences, but we really want to have the longest sequences. So if you sequence in one way and the other way, you can then merge those two reads and get a longer sequence. I don't know if I was clear for that. And with this technology, you can have up to 50 millions and maybe more sequences in one run that last for around 1 day. You need a more step between your PCR, the amplification of your barcode, and the sequencing. You need to add small sequence at the end of your amplicon, PCR amplicon. And why is that? It's because you can have millions of sequence in one run of Illumina, and you don't need this amount of sequence for one sample. Usually, metabarcoding studies have between 50,000 and 150,000 sequence per sample. So we will put multiple sequence in one Illumina run. And to differentiate all those, the sequence to can discriminate that it comes from this sample or this sample, we put some index. So index is just very small piece of DNA, ACTG, for instance, it's index 1, ATTG, it's index 2, and you will have a unique combination for each sample. And also, at these steps, you add things that are mandatory for the Illumina technology, that are adapters to the flow cells and sequencing initiation sites at both ends of your amplicon. And now you are ready for sequencing. So I will get into details Illumina technology. It works with a flow cell. So it's this on the top. And in this flow cell, you have those adapters that we add. You have plenty of them that cover the flow cells. So when you put your mix of samples, your DNA amplicon will hybrid to those adapters. And they will be everywhere in the flow cell. So it's the first step of Illumina sequencing. It's to the fixation of the amplicon in the flow cells. Then the second step is to locally amplify your sequence in a high number using PCR, as we just saw before, and using a thing that is called bridge amplification. That is, your amplicon is fixed here, and then the adapters can hybrid to this one, and then the polymerase hybrid, and you are sure that you amplify locally your sequence. And then you have a cluster of sequences that are identical. And it's the basis of Illumina technology. It's to, and why you do those clusters is that you can now see your cluster using a camera, a very, very powerful camera that you can see those. And so yeah, it's the basis of Illumina sequencing. Then it starts to sequence the first read. So it removes, as after the you make a cluster, you have DNA amplicon in all the ways. So you remove and you only keep the forward strands that start with the adapters P5 and that ends with the adapter P7. And then, so all the strands of DNA are in the same direction. And then the sequencing technology use nucleotides that are fluorescent, and each nucleotide will have a different color of fluorescence. And they have a reversible terminator, so when they are added because they are complementary to the sequence, they will flash. And as you have clusters, you can see a blue flash or a red flash, either if you incorporate a or a T. And it will do that. And the camera will monitor, record the information for all the clusters you have in the flow cells. So the millions of reads you have in the flow cells. And then at one position, you will have a flash of a green flash and a blue flash and a red flash and a green flash, etc. And it's how your DNA is sequenced. It is also at this step that you sequence your index. And then you do the same thing, but for the reverse strand. And so you got another, the read 2 of your amplicon with 2 index. And those 2 index are used for what is called demultiplexing. That is, and it is often the sequencing platform that will do that. It's just to put back the sequence in the sample they belong. And it's often done by the sequencing platform. And now you have nice text files with all your sequences. I will get into detail of what do they look like. And maybe you have questions on the Illumina technology before.
Sorry, can you explain the function of index again?
Yeah. It's because in one flow cell, you can sequence millions of sequence using one flow cell. And it's a too large amount of sequence for only one sample. So you will put multiple samples in one single flow cell. And to distinguish the sequence, this one belongs to sample 1, this one belongs to sample 2, this one belongs to sample 3. We add this small sequence that are called index. And this index will be specific to one sample. Now it can be another one, and then the combination of those two are like, I don't know precisely how many indexes you have, but there are really a lot of them. And for instances, you will have index 1, which has a specific sequence, and in the index 2, that has also another specific sequence. And all the reads, all the amplicons that will have index 1 and index 2 belongs to this sample. And it's how it's done. And reads from another sample will have index 1 and index 3, for instance. And then you can say, okay, they are from this sample. Yeah, it's just label. It's
combination of sticky notes in the DNA world. No, I don't need it. It's okay.
That was a very good question. And I was thinking when we take our samples to your lab, this means that 4 samples from Sava Lake and 4 samples from Plava Banja Lake. So we have 8 samples. So these 4 samples will be in a group where you put, for example, 2 indices. Yes, and 4 samples with Plava Banja will have maybe 3 indices or something that we decide just to be different from the Sava Lake. So 8 samples will be sent together in the same run, but we will distinguish them because Sava Lakes will have exactly known 2 indices and Plava Banja will have 3 indices. Yeah. So that's the reason why we are putting indices.
And more precisely, it's not, you will not distinguish the different lakes, you will distinguish each sample of your lake. Each sample.
So yes, so the Sava Lake sample 1 will have
a particular
indices combination, and the Sava Lake number 2 will have, and so on and so on. Okay.
And you can have up to 300. I don't know the runs we use, but it's
Yes, in fact, when the platform we used to use has developed a way to separate 380 samples with different indices, and so it's really the sequence of this index that will, the combination of letters of this index that will help you to identify your sample. And when you have all these sequences together, then you will select, filter your samples by these index sequences and say, okay, all these have the same index sequences, so they belong, we know they belong to this sample, and all these others have another sequence, and that's what we call the multiplexing. That you will have your, oh, sorry, you will have your, oh, sorry, you will have your, sorry. You will have your your samples separate by your index, and you will work with each data set, one data set per sample, and really it's very straightforward to do.
So the reason is financial. Otherwise, or in the past, we would sample a sequence one by one. In this way, we can put them together and send as a one run and pay one run. So 380 samples can be sequenced together per the cost of one run, if I understood.
And also it's because you don't need 50 millions of reads for one sample to assess the diversity and to have all the barcodes. You need fewer sequences, and you don't need to sequence that much that one run can do. That is why. But yes, it's more. It's lowers the cost.
Yeah. Thank you.
Yes, sorry. In fact, for sure, there are some terms, some words that we use, and for us, they seems to be quite usual to use, like multiplexing, demultiplexing. We use the term you will see after dereplication, and sometimes we forget to define these words. So please tell us, I don't understand what you call multiplexing. So multiplexing is when we have several samples from your different lakes and different season, but we multiplex together and we send to a single sequencing run. And after that, we have to demultiplex thanks to these different indices, which are different from a sample to another. Okay? Do you have other questions?
The first step in data analysis then.
Sorry.
The multiplexing is then the first step to data analysis when you get the. Yeah.
It is, and often it is done by the sequencing platform. So it's not something you have to deal, but sometimes you have to deal with that. But it's not, it's not in the majority of cases.
No questions in the back. I can see some people sleeping there. Kalman, what are you doing? You have a question? Oh, great.
Yeah, I was just wondering that, okay, you said that for one sample, maybe, I don't know, 100,000 reads is optimal, right?
Sorry, I did not.
The reads per sample. You said 50,000 or 100,000. And what if, for example, I want to focus on a very rare species in a community? Would you consider to increase this number or
yes, that? Yes. This can be increased indeed if you have questions, and particularly if you are interested in rare and not abundant species in your sample. You can increase because it's you that choose the number of you choose the sequencing depths, and in some cases you can ask, oh, okay, I want this amount of sequence per sample, and that could be interesting for rare species.
Okay, thank you.
So now we are leaving the lab and we are going into back to the computer. So at the end of the sequencing platform sends you what is called FASTQ files. Those files looks like this. They are files that contains your sequence and the quality of each base of your sequence. So it's really a text file where here you have what is called the def line that starts with I don't know, arrobas, and with an at, with some information. And then you have your first sequence. And then you have a plus to distinguish your sequence to the quality score of each base of your sequence. And then you have the next sequence. And for samples where you will have hundreds, thousands of sequence, you will have this hundreds, thousands of time. The def line, just for information, contains information regarding the Illumina machine that was used, the flow cells, and you will also have the position of the cluster in the flow cell. And this information is used for merging read 1 and read 2 because they were sequenced in the same cluster. Because actually you will have two files because we sequence in one way and the other way. So you will have, for one sample, you will have a file reads that were sequenced in the other way. And each time with hundreds, thousands of sequence in it. So of course, you cannot analyze this using Excel or other things because it will take years. So you go through bioinformatic pipelines. And the object are, during those steps, you will manage to treat all these large amount of information that are millions of reads, but also it is at these steps that you correct the reads from the PCR and sequencing errors. You try to reconstruct groups of sequence that will be equivalent as species. I will explain this a little bit. So the three main bioinformatics steps is first to get complete barcode that are the merging of read 1 and read 2 and then have the complete sequence of high quality. Then you reconstruct your species and then you assign a taxonomy to those species. So the first step, you just look at the quality profile of the reads that are in the FASTQ files. And if they are low profile, you remove the reads. Then at these steps, you also remove the primers because you sequenced them during the sequencing. So it's not truly part of the sequence. So you removed your primers. And then you merge the reads to reconstruct the full barcode sequence you want. And it is also at these steps that you remove camera that you created during the PCR, which is an unavoidable thing that occur. And then at the end, you have sequence of good quality without camera. And you can go to the next step, which is to reconstruct species. And why we do that? Why don't we consider each sequence to be one species and we are happy? It's because you have natural intraspecific variability within a species. You will have slight differences. And because of sequencing errors and PCR errors also. So how does it work? I represented it like that. So here you have your sample sequence, the sequence that were present in your sample at the beginning before you get through all the pipelines, the Metabarcoding workflow. So each dot represents a sequence and each size represents its abundance in your sample. After PCR and sequencing, you will have small variants of this sequence that are produced, but that are in reality the species that are just a variation of the original sequence. So one way to deal with that is to cluster your reads. You say, okay, all the reads that have 98% identity, I group them and I say it's a kind of species, what we called OTUs, for operational taxonomic units. So you take those. All those sequences are identical at 97%, and you say, okay, it's one species. Well, this one also, this one also. But sometimes you can put two different species together that have less variation than 97%, for instance. And as it is a hard threshold, I don't know if you cannot deal with the natural variability between different species. So there are also another way is to use soft thresholds, and other algorithms developed that, like Swarm. But I won't get into much more detail than this. And another solution that is interesting and that appeared after this clustering way to reconstruct species, it's to denoise your sequence, just to try to find the sequencing errors and correct your sequence from those sequencing errors and get back to the true sequence that was present at the beginning. And that can be done with Dada2. And I will discuss of this a little bit later. So maybe you have some question for this part? Yes.
What is Chimera?
Chimera?
Yeah.
It's during the PCR, if we go back. It's when the DNA polymerase stopped for some reason, and this little part that stopped, where you stop the amplification, can hybrid to the wrong template of another species. And it's because you have multiple different sequence, different species in your sample at the beginning. The strands. So you will have a sequence where you have the first part is from this species, and the second part is the sequence of this species. And this is not a true sequence that is present in your sample at the beginning. It's an artificial sequence. So you want to remove it. So it's done by algorithm, and they basically look at a sequence that have that are similar to one sequence and similar to other sequence. If you find that they are identical to two different sequences, they remove it.
Identity. When we talk about sequence identity, is it clear for everybody? Is it clear for you? Yeah. Okay. We have an online question. Okay.
So Patricia Naik from India, why do we need adapters to in NGS? Why do we need adapters to in NGS?
Oh, it's because the technology works with a flow cell where you already have those adapters, and then you want to kind of fix your sequence in those adapters, and then will be present all over the flow cell. It's the way Illumina technology works.
And one more difference between positive and negative controls.
Ah, yeah, it's an interesting question. So negative controls are used to check if you don't have, for instance, DNA in your regions for DNA extraction. It's to check if everything was free, everything you want to be free of DNA is free of DNA. And positive controls is because sometimes, and particularly the polymerase chain reaction can be complex. And as you start from an environmental samples, you can have still stuff from these environmental samples, and the DNA polymerase won't work well and everything. So positive control is to see, okay, I did everything well for my PCR. It's just my template that is that has something wrong, but I can amplify. I did it. As you don't see what you put in your tube, you want to be sure you put everything that is needed for the PCR. For instance, if you don't have any bend at the end, it might be for many reasons. It might be because you forgot to put DNA polymerase. It can be of many reasons. So if you put a positive control, you will be sure you did everything well. And if your other samples doesn't work, it's because of maybe the sample itself. Any other questions?
Oh, you said sequence identity and you stopped.
It's Frederick that was asking me.
You said sequence identity.
Yes, basically, when we talk about sequence identity, well, you need to make an alignment of your sequences. So you put the same position of, for instance, two species which have been this sequence. You put them, you align them. You put the same position of each codon on the same on the same column, and then you look if each sequence has the same codon, has the same nucleotide, and then for a certain distance or for a certain stretch, you consider, okay, this species has two different nucleotide difference. And it's a way to calculate the distance between two sequences. So we talk about identity, but we could talk about genetic distance. So why were you talking about identity? I don't remember.
No,
But
it's you that.
No, because you
yes.
yes, because you use this term maybe here.
It's for the clustering.
Yes, it was for the clustering. Yeah, sorry. Yeah, sorry. Yes, for instance, so when you consider an identity of 99%, it means that 99% of nucleotide must be the same between the two sequences. Yeah.
I just wanted to ask this clustering at the end, it actually depends from your expertise. You decide at that moment this belongs to one cluster or to another, or program decides, or how it goes, the 96 or 99%, or what to consider a species good to ask.
So often we use the very common thresholds, and one is 97% identity, for instance, because it was thought to be representative of species, particularly for bacteria, and now we know it is not. But yeah, it's that's why we developed other way to cluster that are not using one identity. And for this clustering, you don't give the algorithm, okay, I want this threshold. It decides itself depending on the sequence. It's automatically done by the algorithm. And it's why also it's very interesting, the denoising. It's also you don't choose it's not dependent of the person. You don't choose, and it's the algorithms that do it by itself. Yeah.
Thank you.
Can you go back a little to sequencing? Wait.
Back after.
Forward. Back. Back. Stop. This colorful picture. What is it?
These colors.
Yeah.
It's when you during the sequencing technology works, you have your flow cell, and then they add all the regions to as for PCR to replicate your DNA. So you have nucleotides but you don't have normal nucleotides. You have nucleotides that fluoresce when they will be incorporated in the molecule.
And that's not D-deoxynucleotides.
Yes, it's kind of D-deoxynucleotides, but they have this fluorescence capacity. It's just that, yeah. And then, because you have a cluster, when it flash, you will have this flash of red, and you know that red is an A.
Thank you.
Maybe we can move toward the end. So, and then you assign taxonomically. I want the principle is to compare your sequence to reference databases. I won't go into details into the algorithms under this, but you have to know that you don't fully align your sequence because you have to manage millions of reads, and it will take too much memory and time for, and it's highly very long. So all of these steps compose a bioinformatic workflow, because you have to deal with millions of reads and thousands of OTU, for instance. So you need to use bioinformatics, and it is not an option, or you will pass years analyzing your data. So you have, it's not very an easy part for everyone, but you have different softwares that are available to do all this bioinformatic analysis. And I gave some examples here, and they rely on programming languages, but it's not that bad. And for instance, I will speak more of Dada2, the denoising algorithm that use, I don't know if some of you know R, which is a very famous language for biological, ecological, and statistical analysis. So this workflow Dada2 works with this language. So if you're already using this language for ecological analysis, you should be able to analyze your sequence quite easily. So the Dada2 pipeline presents all the steps you need to analyze your sequence. So first, it filters the quality of your sequence. It does the denoising. It merges your reads. Then you have your ASV table. Then you remove Chimera, and then you can assign taxonomy. And this is all done by this pipeline. And what is cool is that you have an online tutorial that explains all the steps for your analysis, all the commands you have to use to analyze your data. And also, as it is using R, it is a widely used language for biologists, and most of the information you are looking for are in the internet. You will find it on the internet, because there is a great community of biologists that are helping each other on the internet. So I hope you are not like this guy after these 2 hours of talks. But what I want to to I will finish my talk with this, is that each steps can have an influence on your final results. And for instance, when you sample phytoplankton in freshwater, you will also sample heterotrophic bacteria you are not interested in. If you do not optimize your DNA extraction, maybe you won't extract DNA from all the species you're interested in, you will miss some. If you, and it's the part that is worse, if you don't optimize your barcode, you can have heterotrophic bacterial sequence at the end, and you might not see all the sequence of the phytoplankton. It is also at these steps that you will produce chimera that are not real sequences. Then during the sequencing, you make some sequencing errors. And then if you don't deal with that with your bioinformatic analysis, you will end with reads. So you have reads, but they are not representative of what you want at the beginning. So it's really important to optimize all your steps to be sure that you have reliable results at the end. Even if you don't have 100% of the phytoplankton community, you'll still have reliable results at the end. And you will see in the next presentation of how this workflow was designed, optimized, tested, verified for the biomonitoring of diatoms, and how we are currently doing this for the biomonitoring of phytoplankton. So thank you for your attention. If you have any questions, or maybe you need a coffee break. It's not, oh, I'm sorry. Okay.
I have more like a comment because I was confused in many steps when you say you do it, you, and I was like, I'm gonna watch this glowing thing. So Daniela shortly explained machine does it. So most of these things are actually just explaining the steps that are in practice simple. Basically, you're using the steps in machines and everything is done by itself. This is just for us to know what is behind the processes and why it's done and so on. So that's why I was so sometimes in your images we are not sure what is the machine, what is in the computer, what is on the, so it's a computer, so what is just a representation of something that is in the sample. So it's,
basically for the sequencing, you send your samples and they do everything, and then you get the sequence.
Okay, thank you.
Thank you.